We've talked quite a bit about neurotransmitter receptors that are ion channels and open in response to binding by their neurotransmitter. But before we can discuss how synaptic weights can change, we need to introduce another type of receptor. And this type, called the G-protein receptor, is quite different in the way it affects its neuron. And also, once again, there are lots of different types of G-protein receptors, all with different effects. G-protein receptors are also embedded in the neuron membrane, often in the membrane of a postsynaptic neuron next to the synaptic cleft, much like ion channel receptors. G-protein receptors all have a structure that passes through the membrane 7 times, as I've illustrated conceptually. Outside the neuron, they have receptor sites that are able to bond to one specific neurotransmitter. On the inside of the membrane and bound to the membrane, there are G-proteins that interact with the receptors. G-proteins have 3 components, alpha, beta, and gamma, that in the inactive state are all bound together. The alpha component is also bound to a chemical called guanosine diphosphate, or GDP. And GDP has 2 phosphate groups. And within the neuron, there are molecules of the higher energy guanosine triphosphate, or GTP, with 3 phosphate groups. There's an enzyme which is also bound to the inside of the membrane, which can be activated by the G-protein if the G-protein is itself activated. And finally, there are molecules within the neuron environment which can in turn be converted by the enzyme to a different molecule that activates various chemical processes in the neuron. This different molecule is called a second messenger. And in this terminology, the external neurotransmitter is the first messenger, carrying information to the neuron, and the second messenger molecule carries the information on within the neuron. If the neurotransmitter binds to the receptor protein, a specific region of the receptor is activated. The G-protein is free to move around on the cell wall and binds to the activated region. This binding makes the G-protein complex open to substitution of the GDP by GTP. When the GTP replaces the GDP, the G-protein breaks apart, revealing 2 activated regions. The activated region on the alpha component can then activate the enzyme. The enzyme configuration is changed by this activation to give it a receptor site for the inactive second messenger molecules. The G-protein alpha segment can activate a number of enzyme molecules. And the activated enzyme can convert locally available molecules into active second messenger molecules, which are then released into the cell. Multiple second messenger molecules can be generated by a single activated enzyme. So there's a considerable amplification of the single neurotransmitter molecule signal. Now, the G-protein mechanisms are a bit slower than direct ion channel mechanisms, but can result in long-term structural changes to the target neuron. And I haven't really talked about the second activated site within the G-protein after it's been split, but it can also trigger chemical reactions that we'll touch on a little bit later in the course. Now, there are lots of different molecules that can act as neurotransmitters. In other words, there are receptors for these molecules that influence the interior of the neuron. Some neurotransmitters are relatively simple molecules like amino acids, and the most common neurotransmitters in the brain are glutamate which is excitatory, and GABA, which is inhibitory. Monoamine neurotransmitters are slightly larger molecules, and often the neurons that generate these neurotransmitters are all located in one or a small number of compact anatomical structures in the brain, but target a wide range of other structures throughout the brain. Neuropeptides are chains of amino acids, in other words, very small proteins, and at least 50 peptides are known that act as neurotransmitter. Finally, some very small molecules like zinc ions or nitric oxide can also act somewhat like neurotransmitters. In general, a neurotransmitter releases one neurotransmitter or sometimes two or three neurotransmitters, and it's the same neurotransmitter or neurotransmitters to all its targets. A neuron that delivers mainly glutamate to all its targets is called a glutamergic neuron. A neuron that delivers GABA is called GABAergic. A neuron that delivers acetylcholine is called cholinergic, and so on. If a neuron delivers small quantities of other neurotransmitters along with the primary neurotransmitter, those other neurotransmitters are packaged generally in the same vesicle and they're released simultaneously. But any one neurotransmitter can produce different effects on a target neuron because the effect of a neurotransmitter depends on what receptors are present. And there are generally lots of different receptors for any one neurotransmitter. And as I described earlier, G-protein receptors generally exert their effects through second messengers, where the external neurotransmitter is the first messenger. Different types of G-protein activate different second messengers, but the main second messengers are actually these 5. And the second messengers exert complex effects on chemical processes in the neuron that maintain its structure. So, in order to understand what's going on, we'll first have to outline the structural chemical processes.